Hi all, my name is Anthony Valente and I am part of the University of Washington's Department of Genome Sciences, and today I will be talking about Phosphopedia 2.0, a modern targeted phosphoproteomics database. For years, discovery mass spec methods which seek to characterize the diversity of peptides in a sample have become the workhorses of phosphoproteomics. Methods such as data-dependent acquisition can analyze tens of thousands of peptides per run and can be combined with fractionation to provide deep analyses of samples. However, these methods are not intrinsically set up for reproducible quantification. In many cases, what we want to measure only makes up a slim amount of the detections, such as a single signaling pathway. Here, the stochasticity of data-dependent acquisition can adversely affect our downstream analysis by causing missing data across our experiments. Instead, targeted methods such as parallel reaction monitoring and data-independent acquisition provide us with the ability to go after only what we care about. Often these methods boast high sensitivity and theoretically provide the ability to get 100% reproducibility. The caveat is that they can require a lot of upfront knowledge about peptides of interest, which may be difficult or time-consuming to generate. In order to facilitate these kinds of assays, in 2016 the VN Lab distilled nearly 1,000 runs, representing hundreds of hours of mass spec time, into Phosphopedia, a community resource for targeted phosphoproteomics. One limitation this database continues to have is that while our peptides boast high numbers of PSMs, the number of matches per peptide is heavily skewed. Most peptides within the database have 5 or less matches, limiting the quality of the information. However, while internal and public data has continued to grow, Phosphopedia has remained static. Therefore, what I'll talk to you about today is our attempts to inject more information into Phosphopedia with automated data analysis, as well as our attempts to move beyond the necessities of peptide detection with predictions for modeling. In order to address Phosphopedia's data needs, we built a fully automated pipeline powered by the SnakeMake workflow management system. This has allowed us to build a highly portable system entirely with open source modules that automatically distributes file processing and is able to selectively run only essential components during updates. The pipeline performs data retrieval using the PRIDE RESTful API, requiring only the specification of accession numbers. We then utilize Crux to perform Comet database search and percolator PSM scoring for each run with instrument-specific parameters. PTM localization is scored using PyA-score, our fast in-house version of the A-score algorithm. And finally, FDR is calculated for phospho-PSM, phosphopeptide, and phosphosite level globally across the database. In analyzing the 1,000 files which were part of the original Phosphopedia, our pipeline only experiences a marginal drop in detections. Many of these can be attributed to higher stringency in our new pipeline due to an increased understanding of PSM and detection scoring that we have gained in recent years. With the new database update, we focused on adding nearly 1,000 new internal files, as well as another 1,500 files from external PRIDE accessions. Much of this new external data was also chosen to emphasize diversity in treatments not originally present in Phosphopedia, as well as new tissues such as hepatocytes and colorectal cells. This data garnered 15 million new PSMs for the database, but interestingly enough, only gave us about a 20% increase in phosphopeptide detections over the original data and a 15% increase in phosphosites. This really emphasizes the hard-won gains in information that we get at this level of data. However, looking at what this increase in data means for the new pipeline can give a sense of why it's important to keep up this data ingestion. We really see experimental data as the standard for information within the database, so the real benefit of increasing runs is the increase in detections for many peptides. Here we show the number of PSMs for each peptide in our reanalysis of the original data compared to the addition of 2,500 runs from this new update. While nearly 50% of peptides gained new matches with the additional data, many peptides with low numbers of detections in the original data set are now represented by hundreds of new matches. This gives us much more data to synthesize for these peptides and gives users much higher confidence predictions for information that relies on spectral counts, such as charge state distributions, which I will talk about shortly. But as I said before, we want to go beyond peptides detected in empirical data. And to do this, we've turned to modern methods in peptide property prediction. The results I'll show in the second part of the talk largely come from data from the original Phosphopedia, but we only expect them to improve as we clean the new update. We became excited about data imputation quite quickly when we realized even simple models could provide us with benefit over heuristics, such as in the case of max charge state prediction. We currently supply this information to users of Phosphopedia in the form of spectral counts for charges 2 through 4, but moving beyond this would be ideal for low detection in absent peptides. The classic method to heuristically determine this property is to count charge acceptors, such as basic residues and the N-terminus. Instead, we can explicitly model the charge state distribution given the count of all amino acids using straightforward methods such as logistic regression. This greatly outperforms counting the charge acceptors alone at predicting the max charge state and is simple to include alongside our spectral counts. But with the diversity of peptides within Phosphopedia, we're also able to build much more complex models. For example, recurrent neural networks have become one of the standards for retention time prediction in recent years. However, it is often difficult to get enough high-confidence phosphoproteomic data to train these complex models. The wealth of data within Phosphopedia allows us to filter for the highest confidence peptides and train models which obtain small windows of index retention time error and high correlations, with most outliers being concentrated at higher IRTs, where the gradients are more difficult to align. While this can be used on its own to get IRT predictions for individual peptides, the models can also be easily recalibrated to even small runs, simulated here by a set of mouse brain phosphopeptide samples. Here, recalibration takes less than a minute, even on a CPU, and recalibrated predictions can obtain retention time windows that are minutes smaller than retraining from scratch. What I've detailed today is an effort to continue to synthesize public phosphoproteomic data into a database that can be used by the community. We've built an automated system allowing us to continue updates as new data is published. In addition, recognizing the limits of data-dependent acquisition to detect all peptides that might be of interest in targeted phosphoproteomics, we begun to move our database beyond the necessities of peptide detection, utilizing models for peptide properties. As we move forward, we want to continue to provide predictors for properties that might be of interest. We already have models that work well for miscleavage prediction, and we're excited to be able to apply our 20 million PSMs to build models for fragmentation as well. In building the pipeline, we've also emphasized generality, making it simple to obtain similar databases for other post-translational modifications, such as acetylation and methylation, as well as new model organisms such as mouse and yeast. This is exciting for us because we feel this is the best way to continue to obtain peptide diversity to enhance the quality of our models. With that, I would like to thank the VM Lab for continuing to push me to get more out of their data, as well as the Noble Lab for their mentorship in computational mass spec. This work was funded by R&T grants from the National Institute of Health, and I thank you for your time.